Okay, our next speaker is Sean Prigge from Johns Hopkins. And I guess we need to, change the...
Okay, all right. And thank you to the organizers for trusting me with the podium. So I can tell you today about our efforts to unravel the secrets of the Apicoplast organelle in malaria parasites. So before I dive into our observations, I really wanted to get everybody on the same page about certain key features of the Apicoplast organelle. And so one of them is that it is an organelle that contains a genome. And so it's put here as an observation that was made over 30 years ago now, that the parasites contain 3 genomes. In addition to a large nuclear genome, there are 2 organellar genomes. And so there's a small mitochondrial genome, and then this circular DNA that contains 68 genes that was ultimately mapped back to this organelle that was named the plastid, or the apicoplast. And so that's one thing to remember, is that this is an organelle that contains a genome. And the other is this odd root, this secondary endosymbiotic root that's hypothesized for this organelle. So it's hypothesized to originally come from the prokaryotic world, like the mitochondrion, and originally have been a light-harvesting cyanobacterium that was taken up by a progenitor cell to form an algal cell. And in this state, it would have functioned as a classic 2-membrane light-harvesting chloroplast. And then that entire cell, the algal cell, was then taken up by a progenitor cell, acquiring more membranes in the process. And what we see today is an organelle that contains 4 membranes and still has this remnant organelle harkening back to the prokaryotic world. So it's this prokaryotic connection that really defines a lot of the interesting biology about the apicoplast and some of its vulnerabilities. So that's the apicoplast. Now in terms of what does it do, when we look in this organellar genome, there's really just not much here to tell us what kind of biochemical pathways might exist in this organelle. These 68 genes are largely the genes that you would need to do translation and some housekeeping operations. And really the only hint at metabolism here is this one gene called SUFB. And this is a gene that's predicted to be part of a biosynthetic pathway for making cofactors, enzyme cofactors called iron sulfur clusters. That's really the only hint you get from the apicoplast genome itself, is that there may be the ability to make these clusters. I'm going to come back to this gene in the end and its existence in the apicoplast genome, because I think this provides some hypotheses about some of the behaviors we've observed for the apicoplast as an organelle. So what does it do then? So it turns out it does a lot. So over time, it's hypothesized that a lot of the genes that may have come in this genome before it was so heavily reduced have migrated into the nuclear genome over time through lateral gene transfer. And this is thought to be a large number. So 250 to 500 nuclear encoded proteins are thought to be trafficked back to the apicoplast. To put that in perspective, that'd be 5 to 10% of the genome of the parasite is thought to encode apicoplast proteins. And they're trafficked back, at least the characterized route, is through the secretory pathways. These are secretory proteins. They have a signal peptide. They're translated in the rough ER, packaged into vesicles, and then they fuse with the apicoplast organelle, and that's how proteins are delivered to the organelles through the secretory pathway, and that's also important. So these are major pathways that have been studied over the past 2 decades, and with a lot of work establishing the functions of these, the enzymes that flesh out these pathways. So it's known that the organelle is the site of 2 carbon-intensive pathways, fatty acid biosynthesis and isoprenoid biosynthesis, both of which require iron-sulfur clusters. And that the carbon source is largely phosphoenol pyruvate, a glycolytic intermediate from the cytosol that's taken in, and it's pyruvate that's used to drive these pathways. And there's also part of a heme synthesis pathway here, so it connects back to Dan's talk. A little bit of heme synthesis here. But the major pathway and the most interesting one, the one that's attracted the most attention in recent years, is the isoprenoid synthesis pathway. And so this produces a building block called IPP, which I'll show in the next slide, and is thought to be responsible for essential isoprenoids that exist in different compartments of the parasite cell. And so the first indication that this was an important pathway came over 2 decades ago, when it was observed that the inhibitor, a very specific inhibitor called phosmidomycin, of prokaryotic type of isoprenoid pathways kills parasites. And the products that are being inhibited here are this IPP, isopentenyl pyrophosphate. As shown here, it's a 5-carbon building block with 2 phosphates on it. And a regioisomer called DMAP. So it's really the position of this double bond that differentiates one from the other. Both are produced by the pathway. And these would then be the building blocks which are presumably exported and used by the parasite to build up different isoprenoid products like prenyl tails for proteins, coenzyme Q in the mitochondria, and dolichols for making glycosylation units in the ER, and so on. So this appeared to be an essential pathway based on this work with this inhibitor. And this was really hammered home with work by Ye and Derisi, who a little over a decade ago showed that you can bypass this inhibitor phosmidomycin, bypass the inhibition of this pathway by adding its product back into the culture of the parasite. So if you add a lot of this compound IPP, like 200 micromolar to 400 micromolar of this compound in the parasite media, you can completely bypass the toxic effects of phosmidomycin. And this makes sense because here you're providing the product of the pathway. And so it makes sense that this would allow you to not need the pathway anymore. But what came as a big surprise and was a seminal discovery in this work was you can bypass a lot of other antibiotic drugs which inhibit apicoplast processes with IPP supplementation. And so this gets back to that prokaryotic origin of the apicoplast. It has prokaryotic translation machinery. And so a lot of antibiotics that target bacterial ribosomes like azithromycin, tetracyclines, and so on will kill parasites, and they'll do so by inhibiting these ribosomes specifically in the apicoplast. And it came as a surprise that adding IPP rescues and renders these compounds less toxic. And what the authors showed here is that the IPP is able to keep parasites alive indefinitely for months or even years, actually, in culture. Even though the apicoplast has undergone some kind of radical rearrangement. So if you look in these cells that have been treated with these antibiotics, the single organelle DNA-containing structure of the apicoplast is gone, and you look like you have these vesicles throughout it. So this is called the apicoplast disrupted state. And I'll show a little bit more about what we think is going on in those cells. But it's surprising that the isoprenoid IPP is able to keep these parasites alive. And the implication here, which I'll talk a little bit more about later, is that seems to indicate that IPP and isoprenoids are the only essential product of the organelle. So that makes logical sense, even though we think now that may be wrong. So what we think is going on in this disrupted state is that in the normal state, for instance, if we make a parasite line that's expressing GFP, linked to a trafficking peptide that would enable this protein to be secreted and trafficked back to the parasite apicoplast, what we would have is this fusion protein produced on the rough ER, packaged into vesicles, and then sent to the apicoplast. And of course, other vesicles are sent out through a more constitutive secretory pathway and actually secreted outside the parasite. And so this is what that would look like, a line like that that has GFP in the apicoplast. Just to put, people aren't used to looking at blood stage parasites. This is a red blood cell, and the parasite's kind of in this area here. We see the hemozoan that we were just talking about is this clump here, and the apicoplast is here in green. And in the case where you take these parasites and treat them with an antibiotic like azithromycin, you observe something very different in the parasites. You observe all these puncta. And what we think those are these vesicles that have been produced because, of course, in this disrupted state where we treat with azithromycin, we haven't affected the production of this nuclear encoded protein. So this protein's still presumably produced. The GFP is packaged into vesicles and they're sent off to an apicoplast which no longer exists. And so we think this is the accumulation, what we're seeing in these images, accumulation of these secretory vesicles that don't have a destination. So the organelle's gone, organellar genome's gone, and we have these vesicles throughout the parasite. And surprisingly, these parasites, as I mentioned, can be cultured indefinitely. So this accumulation of vesicles isn't toxic to the parasites. So as a orientation to the work that we've been doing recently in order to dissect what's going on in the apicoplast organelle, this sounds like a really useful tool for doing studies of gene disruption studies to try to look at candidate essential genes that might be important for apicoplast function. And so presumably you can disrupt nuclear encoded genes that would be important for apicoplast function. And even if they're essential, you could keep these parasites alive with this IPP trick, regardless of whether the apicoplast is disrupted or not. And one of the downsides to this method, in addition to the great expense of this compound, I think it's like $30,000 a gram now, so quite expensive. But if you have a lot of money, if you're independently wealthy, there's still a problem, which is this state of the parasite is very, very difficult to cryopreserve. So there's something about the freezing and thawing process which these parasites don't tolerate. And that's kind of a limiting factor for doing genetic studies if you can't save any of the progeny. So one of the things that we did to initiate this work is we looked at a different way to allow these parasites to make these isoprenoid products. And so it's shown here on the right is the sort of pathway that you find in gram-negative bacteria like E. coli and the parasite apicoplast, which is called the MEP pathway for making IPP and DMAP, the two building blocks that are exported from the apicoplast. What we find over here is a different kind of pathway that humans have, totally different enzymes, but they make the same products. And so we considered this part of the pathway, including these 4 enzymes, which convert the compound mevalonate into these same isoprenoid building blocks. And we generated a parasite line that includes 4 enzymes of these types from different sources. But a chimeric pathway that would allow the parasites to, if mevalonate is put into the culture medium, to produce these isoprenoid building blocks, but not in the apicoplast, but in the cytosol. So we express these proteins in the parasite cytosol, and that allows us to add this metabolite mevalonate, which regularly you wouldn't have in parasite media. It's not part of the media recipes. You can add it in or add however much you want. And it allows the parasites to make these isoprenoid building blocks and isoprenoids in the parasite cytosol, which means they no longer need the parasite's MEP pathway that's in the apicoplast. So it's a different kind of bypass. And this bypass solves those problems with cryopreservation and the expense of buying the IPP compound. And so this allows us to do the same sort of experiments where we can conduct genetic experiments, particularly knockout experiments, where we target candidate genes for disruption that might be important for apicoplast function. And we can study these parasites, again, regardless of whether the apicoplast is disrupted or not, as long as we maintain them in mevalonate. So we can determine whether the gene's essential, that'd be whether it's mevalonate dependent or not when we get the knockout line. And we can also look and determine whether the apicoplast has been disrupted or not in these knockout lines. So we can kind of characterize apicoplast function. And so that's actually allowed us to unravel a lot of the secrets of the apicoplast is using this cell line that we call PfMev. But what I want to draw the attention to today is just one of these genes. So this is an expanded view of apicoplast metabolism where we have the same pathways I showed you earlier, like fatty acid synthesis. And we've targeted various genes that are shown in red that are part of these different metabolic pathways. And we've learned a lot in doing that. But the one gene I want to talk about here is this one in yellow. And it's in yellow because it's one of the only gene out of these ones that we attempted to delete that we couldn't delete. So this one was deletion resistant, even in the presence of that mevalonate bypass system. So this is called diphospho-CoA kinase, and it is theoretically responsible for synthesizing coenzyme A, and it's putatively an apicoplast protein. And it makes some sense for it to be there because CoA is needed for this fatty acid biosynthesis pathway. Of course, CoA is needed throughout the cell as well. So, but it's a little suspicious that it's there because the rest of the pathway is in the cytosol. So this is the traditional pathway using this metabolite, which we all put in our in vitro culture medium. So it was discovered in the early 1980s, shortly after the culture system was invented, that this vitamin B5 is an essential metabolite for the parasites. And this is why. Its sole use in the parasites is to synthesize coenzyme A. So this pathway is largely cytosolic, and it's a little suspicious that this fifth enzyme might be in the apicoplast. So the most parsimonious explanation for why we can't knock it out is it's a cytosolic protein, and we can't bypass it because it's not in the apicoplast. So our first job here was to try to determine whether this parasite DPCK is actually in the apicoplast or not. And to do that, we made a parasite line with a second copy of this gene tagged with mCherry as a localization tag. And so in this parasite line where we've knocked in the second copy at a different locus, we have two DPCKs, the native one as well as this tagged mCherry one, which presumably will go to wherever DPCK naturally goes in the parasite. And so when we look at those parasites, we see that the DPCK tagged with mCherry co-localizes quite well with the apicoplast marker that we have, the apicoplast GFP that we have in this line. So there's fairly good co-localization and reasonable coincidence statistics. These are Mander's statistics here down the bottom. And it indicates that the parasite DPCK is indeed an apicoplast-associated protein, presumably in the apicoplast itself. So in this line, what we wanted to do is to be able to then delete the parasite DPCK. And so in order to do that, we tried to bypass it. And the enzyme that we decided to use to bypass is a very well-characterized one. This is the E. coli DPCK. So this is an enzyme whose activity has been established, well-studied, crystal structure, et cetera, et cetera. And so if this enzyme can bypass this candidate parasite one, it actually tells us what the function of the parasite one is. So we generated a parasite line where we appended onto this E. coli DPCK an apicoplast trafficking peptide as well as mCherry, so we can track to make sure it's expressed and where it goes, and created this parasite line that still has the endogenous DPCK, but has in addition this E. coli enzyme, hopefully trafficked to the apicoplast. And in this line, we can see that indeed the E. coli DPCK is expressed, and that it co-localizes as intended with the apicoplast. So it looks like we've successfully sent it to the apicoplast. And this line now allows us to attempt to delete the parasite DPCK. And so we were able to do that in this line. So here's the knock-in line, and here's the knock-in line with the E. coli enzyme, but with the parasite enzyme deleted. So you can see that, so first of all, we succeeded in doing this. In all the attempts where we tried previously to knock out the parasite DPCK, we failed. But in this line, we could knock it out with all the attempts. So the indication here is that the E. coli enzyme appears to functionally complement the parasite DPCK. And again, that's meaningful because we know the activity of the E. coli enzyme. And also, there doesn't seem to be any disruption of the apicoplast organelle. It appears to be a single intact, normal-looking apicoplast. So the problem with calling it quits here is that to really show that the DPCK activity is actually essential for the parasites, we would now want to knock out the E. coli enzyme and show parasite death. And that's not very easy to do in a scenario like this without a conditional system. So what we turned our attention to after this was to generate a conditional expression line for the E. coli DPCK. And so here we use, which I've kind of annotated with a down arrow indicating this is a conditional system. So we use two different systems, one which Dan mentioned in the previous talk, developed by Jacqueline Niles, involves adding aptamers after the gene that you intend to target. That's a knockdown system. I'll describe that in a moment. And the other involves, is specific to the secretory proteins, involves manipulating the trafficking peptide, what we call a conditional localization domain. I'll show what that looks like in the next slide as well. But those are two conditional systems that we can apply to this E. coli gene to be able to control it and remove it later in the experiment. And so the two systems work like this, where normally this conditional localization domain, which is actually a heavily mutated form of the FK506 binding protein, it's mutated to look like an apicoplast trafficking peptide. It's metastable, so it tends to be disordered unless there's a binding ligand here. So when you add a binding ligand to the parasites, which is shield 1, you end up having a very big change in the localization of the protein. So it goes from being an apicoplast localized protein to being in the secretory pathway, and also you see an accumulation in this annular space outside of the parasite, outside of the plasma membrane, but trapped inside the vacuolar membrane. So this is now secreted outside of the cell in a controllable way with this ligand. So that removes it from the apicoplast. And then there's the knockdown system, which Dan alluded to, developed by the Niles lab, where if you remove ATC, you allow this TEDR-DOSI protein to bind to aptamers in the mRNA, which actually causes mRNA degradation and interferes with the translation of the protein. So it's a protein knockdown. So that would be knocking down the levels of the protein produced. So using both of these tools, we were able to show that in the permissive state, the parasites grow with this E. coli, reliant on this E. coli DPCK enzyme. And in the non-permissive state, with both the knockdown and the mislocalization, parasites die. And so this shows that the parasite apicoplast and the DPCK activity in it is essential for parasite survival. And so a big question here is, what happens in the apicoplast disrupted parasites? Is this DPCK activity still active? And so it happens in the cell line. So here I'm showing the same cell line, the one with the conditional expression of the E. coli protein, parasite protein knocked out. And you see after azithromycin treatment, you see that the puncta, the vesicular puncta that exists throughout the cell. So the apicoplast is gone in these cells. And you can confirm that actually by looking at sentinel genes in the 3 genomes of the parasite, the nuclear, apicoplast, and mitochondrial genome. And so in the azithromycin treated line, the sentinel gene for the apicoplast genome is gone compared to the wild type cells, which still have their apicoplast and their apicoplast genomes. This is another indication that this is an apicoplast disrupted parasite line. The apicoplast genome's gone. And in these cells, the important experiment here shows that when we do this permissive versus non-permissive conditions, we find that the DPCK activity is still essential in this apicoplast disrupted state. And so the implication here is that the DPCK activity is still active in this disrupted state. And so this has led us to a model that looks like this, where we have the DPCK activity in the parasite apicoplast producing coenzyme A, which is then used in the organelle, as well as exported to the rest of the cell. And in the apicoplast disrupted state, disrupted with anything, so in this case, the antibiotic azithromycin, the DPCK is still active in the resulting vesicles that exist after apicoplast disruption. And so it's interesting because those vesicles must have some way to import dephospho-CoA, which is a big molecule and quite charged actually, so it wouldn't diffuse into these vesicles without some assistance, nor would CoA be exported without some assistance. And also there must be, kind of indicates that there must be an ATP source. So that indicates that there's a lot of functionality in these apicoplast disrupted vesicles. So they're not really apicoplast minus in the sense that there might be a lot going on actually in these vesicles. And some hint of that comes from transcriptomic and metabolomic studies that we've done. I'm really just showing the results of transcriptomic studies here, where we looked at these mevalonate PfMev parasites prior to and after apicoplast disruption. So shown on the left is a time course, every 8 hours samples for transcriptomic studies. And over here is the same kind of study done in parallel with an apicoplast minus line. And these specifically, this is not the entire parasite genome, these specifically are a collection of known and highly suspected apicoplast genes. These are nuclear genes that are known or highly suspected to encode apicoplast proteins. And so what it appears, given the similarity between the expression profiles, it appears that the timing and the level of expression of apicoplast proteins is pretty much unaltered, regardless of whether the apicoplast is there or not. So the conclusion here is that the nuclear genome really hasn't gotten the message. It doesn't really realize the apicoplast is gone. It carries on producing these proteins as if the apicoplast is still there. And the implication is that a lot of the processes that we associate with the apicoplast organelle are probably still functional in these disrupted apicoplast states, probably still going on in these vesicles. So I want to conclude with the question of, okay, then why do they need isoprenoids? all of the isoprenoid enzymes are encoded in the nuclear genome. So presumably all of the isoprenoid biosynthesis enzymes are in the vesicles after apicoplast disruption. So why don't they make isoprenoids? And I think the answer ultimately comes back to what I mentioned at the beginning, is that the SUFB enzyme is in the apicoplast genome. And so one thing we know that is lost upon apicoplast disruption is the genome. So the SUFB and the apicoplast genome are gone. And the implication is that in this state, these vesicles probably can no longer make iron sulfur clusters. And it turns out that 2 of the enzymes in the isoprenoid pathway rely on iron sulfur clusters. And so you can imagine specifically that the product of IPP and isoprenoid products are lost in the disrupted state. And so really the need to add isoprenoids and the loss of the isoprenoid biosynthesis pathway is really an exception rather than the rule in terms of what's lost in these apicoplast disrupted parasites. And that a lot of other functionalities probably aren't lost. And there may be compounds like coenzyme A that are essential for parasite survival and that are apicoplast products, but they're still being made in these apicoplast disrupted states. So I would like to wrap things up there and thank the people who've worked on this project in my lab, as well as collaborators who've helped with parasitology and also with the transcriptomic and metabolomic studies that we've done on these parasite lines. Okay.
Okay, Ben Sumner, NIH. Do you know or have any suspicion as to why the mevalonate supplemented ones are cryopreservable?
Oh, I think it may have to do with the fact that the amount of mevalonate that you need to add in the medium is quite low. So maybe 10 micromolar, sometimes we'll add up to 50 micromolar. Whereas for the IPP compound, it really doesn't work unless you add hundreds of micromolar. And so this may be something physical. The carbon end of the isoprenoids precursors are kind of hydrophobic. So maybe at those high levels, you're interfering a bit with membranes and that does not survive well upon cryopreservation. And so it may be as simple as that, just lower amount of compound needed.
Thank you.
Very nice talk, Sean. I'm Handrin from Drexel. So these remnants still have essential biochemical functions, such as CoA biosynthesis, right?
Right.
And then you cannot knock out the CoA DPCK enzyme. And if you knock it out, the parasite will die.
Yes. Mm-hmm.
What happened to these remnants? Do they become further fragmented, becomes even smaller?
Oh, so actually we didn't ever really looked at, because of course the parasites are going to die once you get rid of this.
It's a conditional system.
Yeah, in the conditional system, I suppose there's a certain window before the parasites completely die that you could look at these vesicles and try to characterize something about number and size and address your question of whether they're different or further fragmented. And I actually don't know the answer to that. So we haven't looked that closely at that window where we're actually killing the parasites by removing the activity altogether.
Thank
Yeah,
you.
but you could do that.
Up here, sorry. Right at the top. Right at the top. Suyash from the FDA. This was really fascinating. One thing that popped out, so these vesicles that are being sent out from the ER to the apicoplast, I don't know, is it fair to say that they're fully functional apicoplasts? Because they would be still transport proteins required for the actual functioning of the apicoplast once it receives all its proteins. So would it be possible to just express SUFB in the nucleus and have it be transported via the vesicles to see if it becomes a fully functional apicoplast, just not in a singular region of the parasite instead of just,
Yeah, that's a really good suggestion, and people have asked me that before. And the truth is, I've been too scared to do it, so I should probably just do it. But if you look through the plastid genomes of organisms like this, they all retain SUFB. And so it doesn't seem to be an accident. And so my suspicion is there's some reason why it's not easy to transfer SUFB to the nuclear genome and traffic it back. Because all the parasites of this type have retained SUFB and only SUFB in their plastid genome. So I'm guessing there's some difficulty that we would face in doing that. So I've been like too scared to do it. But you never know. I mean, it actually wouldn't be that hard to try. I probably should just try it. That way I can tell people when they ask that I've done it. Yeah. Okay. All right.
One question on Zoom. Given your new insights, can you comment on the processes that lead to Apicoplast organelle loss upon supplementation? Is the implication that it is energetically quite expensive to maintain these organelles?
Did you hear the question?
Yeah, so the question is, I think, whether it's energetically expensive to maintain the organelle and whether it could be lost spontaneously upon IPP supplementation, which kind of would be an indication that it's energetically expensive to maintain it. And it sort of is. So we did one experiment with this mevalonate line where we maintained it for actually a year. And in that state, any apicoplast that's lost just adventitiously can survive. That parasite can survive. And so you do accumulate some apicoplast losses over months, but they seem to come to an equilibrium at some point with about roughly 50% loss of the apicoplast over very long periods of time. And that, I think, provides a hint as to how costly it is to maintain the apicoplast versus how valuable it is to have it. Is the population never goes, even after a year of bypass, to complete loss of the organelle. So that's a hint at the answer to that question. Yes?
Quick question.
He's going to get in trouble.
Are the fragmented apicoplast remnants, are they also four membrane structures, or is there only single membrane?
I have seen a micrograph. It's in my head.
In your head?
A micrograph is in my head. And I must have seen it as a talk because I then went later to look through papers to find the micrograph and I could not find it. So it's either buried in a supplement somewhere in somebody. So I haven't done that. Or I saw it in a talk and it was never published. But I think the answer is that it's a one membrane vesicle and the four membrane structure is lost. Right? I can't point to the publication.
So the
where
whole
that is.
transporter stuff really becomes very interesting, right? If it's
Yes.
single membrane, then you will need to have all the transporter, because the whole idea about having the signal peptide and the transition peptide, that becomes moot in some ways.
Right. And in fact, you could hypothesize that maybe in fact, there's some evidence for this, that the transit peptides are not cut from the proteins once they're in these vesicles
So
properly.
they would not be processed. Right?
The ones that have been examined aren't processed. And so there may be some proteins that aren't active because they need that processing, and many that maybe don't.
Interesting.
Yeah.
Thanks, Sean.
Okay.